Good morning everyone. I Aditya from BSc Chemistry Honors final year is today here to give my audio presentation for the visually impaired students. Topic of my presentation is fermentation. Fermentation is the chemical transformation of organic substances into simpler compounds by the action of enzymes, complex organic catalysts which are produced by microorganisms such as molds, yeasts or bacteria. Fermentation technology is the use of organisms to produce food, pharmaceuticals, and alcoholic beverages on a large scale. The science of fermentation is known as zymology. The principle of fermentation processes. The basic principle involved in the industrial fermentation technology is that organisms are grown under suitable conditions by providing raw materials meeting all the necessary requirements such as nitrogen, salts, trace elements, and vitamins. The end products are formed as the result of their metabolism during the lifespan are released into the media which are extracted for the use by human beings and that have high commercial value. The types based on the respiration. There are two types based on the respiration of the fermentation. First one is aerobic fermentation, which is also known as aerobic glycolysis. It is a metabolic process by which cells metabolize sugar via fermentation in the presence of oxygen and occurs through the repression of the normal respiratory metabolism. Aerobic fermentation means that oxygen is present in the following reaction: wine, beer, and acetic acid vinegar, such as apple cider vinegar, need oxygen in the primary or first stage of fermentation. When creating acetic vinegar, for example, exposing the surface of the vinegar to as much as oxygen as possible, creating a healthy, flavorful vinegar with the correct pH. Anaerobic metabolism fermentation. In this type of fermentation, the oxygen is not so required. There are two types of aerobic respiration. That one is homofermentation when only one type of product is formed, and second one is heterofermentation in which the products more than one are formed. Now see what is lactic acid fermentation. The lactic acid is formed when pyruvate produced in glycolysis and NAD plus is generated from NADH. Enzyme lactate dehydrogenase catalyzes this reaction. The lactate is the dehydrogenation salt of the lactic acid. Lactobacillus bacteria prepare curd from milk by this type of fermentation. During intensive exercise, when oxygen supply is inadequate, muscles derive energy by producing the lactic acid which get accumulated in the cells, causing fatigue. Reaction involved is the oxygen in the presence of lactate dehydrogenase is converted into pyruvate, and then pyruvate is converted into lactate by producing NADH and the in the cyclic process. The second fermentation we should study is alcoholic fermentation. This is used in the industrial production of wine, beer, biofuel, etc., and the end product is alcohol and carbon dioxide. The pyruvic acid breaks down into the acetaldehyde and carbon dioxide is released. In the next step, the ethanol is formed from the acetaldehyde. The NAD plus is also formed from the NADH, which is reused in the glycolysis. Yeast and some bacteria carry out this type of fermentation. Enzyme pyruvic acid decarboxylase and alcohol dehydrogenase catalyze these reactions. In this reaction, the glucose is converted into the pyruvate, and then the pyruvate is converted into the 2-acetylaldehyde, and then this is converted into the ethanol. Now just see the citric acid fermentation. The citric acid having the IUPAC name 2-hydroxypropane 1,2,3 tricarboxylic acid derive its name from the Latin word citrus with a molecular weight of 210.14 gram per mole, which contains 3 carboxyacylic oxalic functional groups with 3 different values of pKa in the essence 3.1, 4.7 and 6.4. Citric acid, a primary metabolic product, is the most important organic acid produced in the large quantity and is extensively used in the food and pharmaceutical industries. Citric acid is a tricarboxylic acid having the chemical structure in the shown in the video. It is formed in the tricarboxylic acid cycle or the Krebs cycle and is found in small quantities in all the living organisms. Citric acid was commercially produced in the England in 1826 from lemons contain 7 to 9% citric acid until 1919. It is produced mainly by the submerged fermentation using the Aspergillus niger or Candida sp. from different sources of carbohydrates such as molasses and starch-based media. Citric acid production. About 99% of the world's citric production occurs through the microbial processes which can be carried out using the submerged or surface culture. The 70% of anhydrous or monohydrate form of citric acid is used in the food and beverage industry as an acidifier or antioxidant to preserve or enhance the flavor and aromas of fruit juices, ice cream, and marmalades. 20% is used in the pharmaceutical industries as antioxidant to prevent vitamins, effervescent, pH corrector, blood preservative, or in the form of iron citrate as a source of iron for the body, as well as in the tablets, ointments, and cosmetic preparations. In the chemical industry, which uses the remaining 10%, is employed as the foaming agent for the softening and treatment of textiles. Now see what is surface fermentation. Using rice bran or other starch or sugar-rich substrate have been used for the surface fermentation by the Aspergillus niger or in the liquid solution in the flat aluminium or stainless steel pans. A niger can be used to produce citric acid despite the high content of the trace metals in the rice bran are used. The citric acid is extracted from the bran by leaching and is then pipetted using the resulting solution as calcium citrate. Now, so what is submerged fermentation? It is a common practice when citric acid is made. The fermenter is made up of acid-resistant material such as stainless steel. The carbohydrate sources are molasses decatenized by the ion exchange, sucrose or glucose, magnesium sulfate, 7 moles of water, and KH2 phosphate at about 1% and 0.05 to 2% respectively are added. The pH is never allowed higher than that of 3.5. Copper is used as up to 50 parts per million as an antagonist of the enzyme aconitase, which requires iron. 1 to 5% of methanol, isopropanol or ethanol when added to the fermentations containing unpurified materials increase the yields are reduced in the media with the purified materials. As high aeration is deleterious to the citric acid production, mechanical agitation is not necessary and air may be bubbled through. Antifoam is added. The fungus occurs as a uniform dispersal of the pellets in the medium and the fermentation lasts for about 5 to 14 days. Thank you.